want to be here, please go ahead and do so. We're starting off right away. We welcome the Provost, sir. Welcome, sir. We welcome the Dean of the Faculty of Basic Medical Sciences. You're welcome, ma. We welcome the Dean of the Faculty of Basic Clinical Sciences. You're welcome, sir. We welcome the Secretary to the College. You're welcome, sir. We welcome the head of the Sparkling Committee, Professor A.M. Adeoye. You're welcome, sir. And we welcome our distinguished Deputy Chairman, Medical Advisory Committee, Professor Olaoye. You're welcome, ma, who is also a member of the Sparkling Committee. We'll start off right away by taking the anthems. BCC, we're set to go. Shall we rise for the anthems?
Nigeria, we hail thee, our own dear native land. Though tribe and tongue may differ, in brotherhood we stand. Nigerians old and proud to serve our sovereign motherland.
Thank you. We may be seated right away, saving time and hoping we can catch up with the lost few minutes. You will please welcome with me Professor Abiodun Adeoye, cardiologist of international repute, who heads the special committee in the College of Medicine that is hosting today's event. A round of applause. You're welcome, sir.
Our dear Provost of our college, chairman of this, Deputy Provost in absentia, Dean of the Faculty, Professor Lawa, and all the other deans here, College Secretary, Faculty Officer, all the members of the Sparkling Committee, gentlemen, why the Dean will give welcome address, Leo, produce this audience, Sparkling, responsible for strategic collaborations, partnership, advancement, research, and as well as grant. So today we begin. One of our mandates is to organize faculty lecture, special guest lecture. Just Dr. Ojesino that is here, who happens to be an alumnus of this place, we are privileged to have you to our midst, and we know he's here to do justice today. And we want to give special thanks to Provost Akemi for connecting us with him. He will also be giving, among others, and that he has, will be giving a grand round Department of Medicine at 4 o'clock. As a Sparkling Committee, we have a series of faculty lectures and special lecture lineup for us. And we will be glad to have you around once we call upon you. I want to thank everyone for coming for this event. Fasten your seatbelt. The plane is about to take off.
Thank you very much, sir. In that short speech, he has told us what Sparkling stands for, and I hope that we have noted it. But among other things, it stands for strategic partnership, research, innovation, and these are key to what we need in this present age and time. Please welcome with me the indefatigable, fantastic, hardworking, diligent Dean of the Faculty of the Clinical Sciences, Professor T.A. Lawal, to give his address.
Thank you so much, brother. The Provost of the College of Medicine, the Deputy Registrar and Secretary to the College of Medicine, Dean Faculty of Basic Medical Sciences, all our professors from the College of Medicine and beyond, heads of departments and the faculty and other faculties, the guest lecturer, our sub-deans, the faculty officer, staff, and our ever versatile students, distinguished ladies and gentlemen. Faculty lectures were introduced to afford everyone the opportunity to listen to aspects of research efforts that will benefit both staff and students. The Faculty of Clinical Sciences, and indeed the College of Medicine of the University of Ibadan, is blessed with great researchers who have continued to make impact on our society. We have had a good run of these lectures in our faculty. However, there had been a hiatus over the past few years, partly due to the COVID-19 pandemic. It is a great pleasure that our hard-working Committee on Strategic Partnerships, Adjunct Professorship, Research, Sparkling Innovation, and Grants, that's the Sparkling Committee, under Professor Abiodun Adeoye, has spearheaded the return of this lecture series. Our faculty lectures are delivered by internal members of the faculty. Often, these lectures are delivered by individuals outside our university, and they become guest lectures as we have today. Today's lecture will be delivered by an alumnus of the university, Dr. Akinyemi Ojesino. He is a big brother, someone who has always excelled right from student days at the University of Ibadan. His work on oncopathogenomics will today illuminate how viruses, such as the human papilloma virus, synergize with genomic alterations to influence cervical cancer initiation, progression, response to therapy, as well as prognosis. This lecture is coming at a very auspicious time. Following the exploits of another one of our own, Oludare Morasembelo, a professor of obstetrics and gynecology, who recently, together with his HPV consortium, welcomed a groundbreaking event at the College of Medicine of the University of Ibadan. We can equally recall that the 9th Dean of the Faculty of Clinical Sciences, Professor Isaac Folorunsho Adewale, is the chairman of the National Task Force on Cervical Cancer Elimination. In fact, a lot of our elders, a lot of our teachers have worked on cervical cancer and human papilloma virus, that Ibadan can be said to be the capital of research on cervical cancer in Nigeria. It is therefore pertinent to note all efforts at understanding the extent of involvement of HPV in human tissues is the right way to go, and Nigeria works to save the future of our men, of our women, pardon. At our courtesy call earlier this morning on the Vice Chancellor of the University of Ibadan, Professor Kayode Adebowale, MNI FAS, he reiterated his support for cross-cutting multidisciplinary approaches to research in our university. This lecture will showcase how working together we can achieve more than ever before. Ladies and gentlemen, on behalf of the students and staff of the Faculty of Clinical Sciences of the College of Medicine, University of Ibadan, I welcome you all to this special guest lecture. Thank you.
Thank you very much, our Dean, sir. Right away, to save our time, we quickly move on to take the opening remarks from the Provost of the College of Medicine, University of Ibadan, the first and the best. Please welcome with me Professor Tio Ogunderon, FAS, to give his opening remarks.
Good afternoon, distinguished members of high table here. Are present. Has been written. On behalf of the College of Medicine, by the Plot Thickens, global insights from both timely. Cervical cancer remains women world. Women diagnosed 1,000 deaths, alarming or in low. 1,000 new cervical cancer, 1,000 deaths, but daughters, yet hope. Science is coming out, helping us unravel how it might. Every discovery worth taking, but with it, new opportunities for earlier detection, more effective, ultimately, college must ensure that such scientific policies bench to the fact. This lecture call to welcome the guest speaker, passion for and for trans. Or Jeshina. Clap, clap. Whose pioneering work in cancer genomics is helping to shape a future community. Let us all get inspired to part right.
Thank you very much, sir. I see that our appetites are already being whet, and we will just move right away to take the citation of the guest lecturer this afternoon. And that will be given by none other than Professor Olushola O. Olawoye, a distinguished ophthalmologist and a deputy CMAC of the University College Hospital. Ibadan, you're welcome, ma'am.
May I ask our distinguished lecturer to stand up? Thank you. From the very beginning, our lecturer today stood out. He has always been an erudite scholar, and you will agree with me as I go through his citation. Dr. Akinyemi Ojeshino obtained his medical training at the College of Medicine, University of Ibadan, with a distinction in biochemistry. Please help me clap for him. And while he was a student, he served as the editor-in-chief of the Dokita Editorial Board. After a stint as a medical research fellow, he has always loved the biological sciences, and he showed so early in his career. After a stint as a medical research fellow at the Institute for Advanced Research and Training at the college, he earned a PhD in biological sciences in public health at Harvard University. Please clap properly. Yes, it's not a mean one. Yes. During his doctoral work at the laboratory of Dr. Phyllis Kanki, he identified molecular determinants of antiretroviral drug resistance in the HIV-1 subtypes prevalent in West Africa. Thereafter, he completed a de Monruyon postdoctoral fellowship under the tutelage of Dr. Matthew Meyerson at the Dana-Farber Cancer Institute and Broad Institute of Harvard and MIT. During this time, he led the first exome and whole genome sequencing-based characterization of cervical cancer and co-developed a computational subtraction tool, PathSec, for pathogen discovery from next-generation sequencing data. He was the first to do this. Please, let's clap for him. He is currently an assistant professor of obstetrics and gynecology at the Medical College of Wisconsin, Milwaukee, USA, where he runs the Ojeshino Oncopathogenomics Laboratory. Along the way, Dr. Ojeshino has received several honors and awards, including, to mention a few, the Rebecca Ridley Cry Fellowship of the Damon Runyon Cancer Research Foundation, multiple AACR GlaxoSmithKline Outstanding Clinical Scholar Awards, the Stuart Scott Memorial Foundation Scholar Award, and wait for this, just wait for this, he was named one of 1,000 inspiring Black scientists in America. He has also gained an international reputation through his leadership of several comprehensive genomic studies on cervical cancer, including the Cancer Genome Atlas, Cervical Cancer Working Group, the NCI HIV Tumor Molecular Characterization Project Working Group on cervical cancer, and the NCI Cervical Cancer Clinical Trials Planning Workshop. Dr. Ojeshino's research is funded by the National Institute of Health, NIH, and lies at the nexus of translational genomics, molecular epidemiology, precision oncology, tumor microbiome, immunotherapy, infectious disease, women's health, and global health. His group investigates the synergy between microbes and genomic alterations in tumor initiation, progression, therapy response, and prognosis. To do this, they combine cutting-edge genomic analysis and functional experiments to interrogate population-based patient samples, laboratory cell lines, and animal models. He has published his work in several prestigious journals, including Nature and Nature Genetics, and has over 28,000 citations with a Scopus H index of 45. Ultimately, this work will have a translational impact by facilitating the development of diagnostic biomarkers and predictive models for early detection, prevention, and prevention of various cancers in local and global contexts. Esteemed and distinguished teachers, senior colleagues, ladies and gentlemen, I present to you an erudite scholar doing great things internationally, putting his feet in the sands of time, excelling all over the world in translational research, and changing the paradigm of cancer research one step at a time. Dr. Akinyemi Ojeshino. Thank you very much.
Good afternoon, everyone. The Provost, the Dean, all of the protocols observed. Thank you for the opportunity to have this conversation with you this afternoon. I want to also recognize, I see many of my teachers in the room and colleagues as well. Great. So I thought I would start today by talking about how proud I am to be an alumnus of this institution. And this okay, good. And this is actually meant for the students. So let me ask you, I guess yesterday I met with Dokita and Cree, and I realized that apparently there was some change. But when I was in medical school, we were taught that Burkitt's lymphoma was the most frequent pediatric cancer in Ibadan. I went through medical school. I saw some Burkitt's lymphoma, but not that many. I was a house officer. I saw some Burkitt's lymphoma, but not that many. So I simply said, is it really true that Burkitt's lymphoma is the most frequent pediatric malignancy in Ibadan? And what did I do? I decided to find out. So I approached Professor Akang of blessed memory about getting access to the Ibadan Cancer Registry. And the result of that investigation was this. This was my very first peer-reviewed article, published because of observations made as a medical student in this institution. And so I want to encourage you that pay attention, be observant, be curious. If you don't get anything else from this talk, know that you can actually make impact because of your observations and things that you do subsequently. I call my lab the Oncopathogenomics lab. It's actually a made-up word. But it basically talks about the fact that we're using a tumor ecology paradigm to look at how microbes and genomic alterations work together in cancer. So our work is in 4 parts. The first, and by the way, I love audience participation, so at some point I'm going to require of you to join me in audience participation. Who's ready for that? Thank you very much. So we do genomic analysis of tumors, focusing primarily on women's cancers. The second thing is that we try to understand atypical cervical cancers. We'll talk about that shortly. So things that are not so common. And then I'm very interested in tumor microbiome and how microbes influence health and disease. And finally, we are also really interested in identifying new microbes that cause cancer or that facilitate cancer. This is probably not new to you. We all know here that the most common tumors in women in Nigeria are breast cancer and cervical cancer, and listed here are some of the risk factors for cervical cancer. So the thing is, we also know, I hope, that most people get infected with HPV at some point or another, but the immune system clears it. And in a small proportion of women, that the virus persists, leading to precancer, and then some of the precancerous lesions proceed to cancer. And you also know that there's a lot, we learn a lot about prevention and how that's important in medicine. And I just thought I should highlight the fact that I think of prevention as everything is prevention. So when we talk about prevention, we're usually talking about primary prevention, which is what you do for risk factors. But technically, secondary prevention is what allows for you to prevent the development of disease, even after exposure to the risk factor. And then tertiary prevention is for prevention of complications. And this is knowledge that I hope everyone knows about the fact that there are 2 main subtypes, or two main histological types of cervical cancer: squamous cell carcinomas and adenocarcinomas. I thought I would sort of highlight something as a prelude to something that I'm really looking forward to. I put these 2 pictures here. The first to show Dr. Harald Zohausen, who was the first person to actually put a link between cervical cancer and human papillomaviruses. And I'll come to talk about why that there's an Ibadan link to some of this work on cervical cancer, which you see in a moment. And the 2 gentlemen on the right are actually the ones who helped to develop the HPV vaccines that are now being used. So I thought I would really do that. I thought it was really, really important to highlight. I didn't even know that the dean and the provost were going to talk about some of this. But the University of Ibadan has actually played tremendous roles in the knowledge that we have across Africa, really, about cervical cancer. So I did some searches, and one of the things I found was this paper from the 1960s, published by Professor Lawson, Dr. Nwosu, and Dr. Olafimiyo, showing, I think this was 1964, thereabouts, some of the earliest work about cervical cancer in Nigeria. Then came the 1970s, where people like Professor Oshukoya, Professor Adelusi, Professor Adeleye, and Professor Junaid played tremendous roles as well in opening up work of various kinds in cervical cancer. And I must say at this point, I am probably going to miss some people. So please forgive me if you or your mentor is not mentioned. It's not on purpose, just a quick summary about some of the accomplishments we have in cervical cancer. Then came what I call the era of my teachers. In this slide are really people who taught me in medical school, who had various types of papers. And by the way, these are from PubMed. So I just did cervical cancer Ibadan. And the things that came up are what I'm presenting. So I call this the era of my teachers. And then came the era of my brothers and sisters, brothers and sisters, mostly senior brothers and senior sisters, who also did some work across the board in cervical cancer. And here is where I'm going to probably, and then came, this is a potpourri really of more recent work, and some of the people who have done some of that work as well as more recently in the 2021 and more recent. And like I said, this is a highly incomplete list. But I just wanted to use it to appreciate the work, the tremendous work that has been done in this place on cervical cancer. So here's the thing. I just talked about the fact that exposure can lead to disease. And usually there's a black box. Don't always know how come exposure leads to, just know it does. So, traditional epidemiology doesn't often consider what is inside that box. On the other hand, molecular biology does not always consider what is outside the box. There's a field called molecular epidemiology which does. It looks at both what's within the box and what's outside the box. So part of what I do is molecular epidemiology and other things. I thought I would use that as a model to kind of show this. I kind of talked about it. I thought I removed the slides, but apparently they're here, which means I have too many slides, but we'll figure it out. So I just wanted to show you here, once again, I talked about this, primary prevention, secondary prevention, tertiary prevention, and then there are sort of solutions or things you do with those different levels. For example, vaccination, which is really, really important. And then we get into testing and determining of diagnosis and then treatment, as it were. So my work really focuses on these two areas that I think about as secondary prevention and tertiary prevention. So I just put this here to talk about the fact that the world is sort of, translational medicine has always existed, really, but there's more emphasis on it a little bit more these days. And I think with respect to cancer, it's really been built on these four things. The first thing is that, First thing is that we have, once again, what I like to call biology-driven prevention. Okay? We have more understanding of the molecular pathways, and there are also genomic technologies now that can be used. I'll talk about some of those for profiling tumors. And then finally, we're in an era of targeted therapy. So really, the combination of all of this has served to make the field of translational cancer genomics and translational medicine, really, because you can apply it in cardiology, you can apply it in various other fields, really important. So, I'm going to do something I call Genomics 101, which is to, okay, good. So, when I was a little boy, my father had this book of after-dinner stories. So I read, one of the stories that captivated me was this journalist who basically wrote about a general and called the general this. And the general got really upset. And she said, I'm so sorry. I hope you're seeing what's going on. And then she said, oh, I'm so sorry, I meant this. And the general got really, really furious. And then finally, the third edition came out and she said this. So, battle scared, bottle scarred, battle scarred. These are typos, right? But a single typo can make a difference in how you interpret information. In the same way, I like to think about mutations in DNA as typos. Some typos don't matter as much, but sometimes some typos will change the meaning of a message. In the same way, in a very simplified way, this is how I think about cancer. Very simplified way. It's a result of miscommunication and misinformation that leads to breakdown of law and order. Everything else is detail, right? Miscommunication, misinformation, breakdown of law and order. So all I spend my time doing is trying to understand what is the misinformation, what is the miscommunication, how do we work on this law and order. Everything else is detail. Okay, so in view of that, I thought I'll talk about mutations a little bit. So what I'm showing you here is if we were a body market and you want to come here, this is the most likely route you will take, correct? Correct? Yes. You have to get on, what's this road called? UIUCH road. You've got to get on that road to get here. But if you stay on that road, you will not get here. The journey to the College of Medicine requires two things. You need to get on that road, but you also need to get off that road. In the same way, the journey to cancer can be thought of as a journey of two parts. You have to get, so there are mutations called germline mutations. They're present in every cell of the body. They are passed down from parent to child. While they increase the likelihood of getting cancer, they are not enough for the development of cancer. You need an additional step of somatic mutations. So when people talk about mutations, for the most part, many of us are talking about the germline ones. They are present really in all cells of the body, but somatic mutations are present in the particular tissue where cancer is being formed. Okay? Now, I have to speed up. I thought I would show you some of those types of mutations that occur. So imagine you have a fragment of DNA. We do what's called paired end sequencing. So the orange part you see is actual sequence. The middle portion is empty. But we generate lots of them, and when you do, we pile up, and we're able to see places where you see these types of mutations, where it's A within the normal genome, or it's a C in the particular individual. We call those point mutations. There's a slightly different variant that are called indels, which are insertions and deletions. And then we have some other types of genomic alterations that are called not at the nucleotide level, but at the chromosomal level. We usually call those copy number alterations. And then sometimes you have some alterations that involve the joining of one part of a chromosome with another part of that, the same chromosome or other chromosomes. We call those translocations. And then finally, you can also find pathogen sequences in human samples. There's another kind of, now here comes the participation. There's another part of mutations that I think, or alterations that I think are really interesting and really cool. I call this my Yoruba man's guide to epigenetics. What is this word? Shout it out, please. Yes? Okay, good. Now, let's go through it. So, what is this word? What is this one? What is this? What is this? Which is a name. What is this? Ojo, what is this? Which is something. What is this? Sorry. What is this? Okay. So, here's the thing. Before I talked about changes at the DNA level, sometimes the DNA itself does not change, but it gets marked. And it affects how genes get expressed. This general area is called epigenetics. And so one day I was thinking about it and I thought, wait. Epigenetics is like Yoruba. We put marks. And the marks change what the word is saying. And so once again, misinformation can occur, right? If the wrong marks are put on DNA. Okay, now to data. I had the opportunity to lead, like I said, the first whole exome and whole genome sequencing of cervical cancer. These are the colleagues we worked with at the time. We published this in Nature in 2014. And at the time, what we showed was this list of mutations. I'll be happy to take questions at the end, but I just want to go through this really quickly. So there were some genes that were already known to be mutated in other cancers, but not in cervical cancer. And these two were found for the first time in any cancer at all. So since then, I have focused a bit on this gene called MAPK1 in the lab. And in 3 different landmark studies, we showed that MAPK1 is always mutated amongst a variety of other genes that are mutated. And so we've gone to the bench, and we've examined this mutation in cell lines. And what we did was basically to overexpress that gene in the wild type form and in the mutant form. And we show that the mutant form is associated with increased cellular proliferation in both a cervical cancer cell line as well as a general cell line. And we've also shown, using wound migration assays, that when cells overexpress the mutation, they actually move faster. As you know, migration is one of the properties, or faster migration, one of the properties of cancer. And then we've also done a lot of work on microbial analysis. So I have a question for you. If you have a mixture of metallic paper clips in gray and plastic paper clips in red, and you wanted to separate them, what would you do? Shout it out. You would use a magnet. Thank you. I actually made this slide for my son when he was about 4 years old, and for a while he thought I went to work to play with magnets. But essentially, this principle is something that we use a lot, where when you do genomic sequencing of human tissues, it could be cancer, it could be something else. That if it's driven, if there are microbes that are involved in that process, you will find both DNA and RNA of both the human and the viral. And then what we do is to computationally subtract out what's human and focus on what's not human. And so, for example, and by the way, along the line, I am showing some pictures of people. These are people who have helped along the line, either led projects that I worked on or have worked on projects that and I'm sorry, I won't give all their names, but I have a list of acknowledgements at the end. So, for example, here you would have a human block, tissue block, that has all sorts of things in there. You extract nucleic acids. So we then align to the human genome. Once we've sequenced, you pull out what's human, and then you also go align to microbial genomes, and you pull out what's microbial, and go on to assess that and study what kinds of microbes are there. Find HPV, it's there. You find bacteria if it's there. And then we can go on to then take what's left and it's not map into anything. Some contigs, if you remember your biochemistry, and then we use that to identify novel bacteria or viruses. So I'm really interested in tumor microbiome, and I like to be really broad when I think about what the tumor microbiome is. I like to think of it as the spectrum of microorganisms that are present in or around tumors. And so they could be exogenous or they could be endogenous. Now, some of you may know from your biochemistry that there's a procedure called 16S sequencing because bacteria have 16S DNA from human DNA, which is 18S. So if you find any 16S DNA in human samples, it has to be one non-human and two microorganismal. And so based on that, we really study 16S bacterial DNA, but I'm really interested in studying RNA because I think that when you study RNA, you can find out not just who's there, but what they're doing. You can get information about the function. Now, there are some methods by which you can also get function if you look at just the DNA. The other thing that we're really interested in is that most people, when they study the microbiome, they study the gut microbiome. And although we're interested in the gut microbiome, we're really also interested in the microbiome in the tissues. So we've done a number of studies in that respect. So for example, in cervical cancer, we looked at women who had hysterectomy. Some of them recurred within or had some progression within 2 years, and some of them did not. Then we looked at their tumors and at the microbes we could find there. We identified pro-inflammatory microbes in the tumors of women who had faster recurrence, whereas we saw anti-inflammatory bacteria in the tumors of women who did not recur. So one of the paradigms of HPV causation is that when HPV infects the cells, after a while, if it persists, it integrates into the human genome. Areas where, and based on that, this is early work, showed that, so the way, let me explain. Box plot. There are about 80 tumors. And of those 80 tumors, there is a red circle that represents one tumor, HPV, that has HPV into that gene. Interesting pattern where the tumor that had HPV had the highest expression. Sorry, the tumor that had HPV had the highest expression in Y compared to the other tumor. That was really interesting. And now there are lots of work being done about this to try to understand how and why HPV. I'm going to leave this. Say that in genomics, I think, of cervical cancer. So the first is the well-known HPV integration associated cervical cancer. But we have found out that in some cases, we don't see evidence that HPV actually integrates into the human genome. The virus remains extrachromosomal or episomal. And then more recently, we have shown that sometimes we actually don't see HPV. Okay? And now people initially said, oh, well, it's a diagnostic, lack of proper diagnosis, or perhaps that it was there but went away, etc. More recently, just to say that the vast majority of HPV, vast majority of cervical cancers are still HPV driven. Okay, so what causes cervical cancer? What causes cervical cancer? This is very important. Please, if there's anybody in the press here, what I'm about to say does not negate the fact that HPV causes cervical cancer. Okay, I'm about to talk about how the plot has thickened. Okay, and what we have seen now is that there are these types of HPV. They are rare, 5 to 10%, where we see no evidence or non-substantial evidence of HPV. We call them HPV-independent cervical cancers. And we were one of the first groups to show this. We had this paper, and here are some of the collaborators, where we basically showed, for example, the other thing that happened was, well, I'll get to that in a moment. So one of the things we showed across 3 different cohorts was that the HPV-independent or HPV-undetected cervical cancers were seen in older women. Second thing we saw, which was really, really interesting, was that we saw this phenomenon in both squamous cell carcinomas and adenocarcinomas, unlike what had been thought before. People said, in addition to the fact that diagnosis wasn't right, that it was because it was mostly adenocarcinomas. But we clearly saw squamous cell carcinomas affected. Now, why am I interested in HPV-negative cancers? It's because of this graph. Let us assume that HPV vaccination is successful, and we wipe out all of HPV-related cervical cancer, right? We will have some that the vaccinations will not deal with. And guess what? Right now, we're seeing evidence in every cohort that we have looked at that the ones that are not associated with HPV actually have worse prognosis, right? And that, for me, is why it's important. And I'm going to skip some of the gene mutations, but I think, Professor Gubi, you'll probably be interested in this. One of the reasons why I'm really happy about this work is that the very latest WHO classification of tumors on female genital tumors actually now lists HPV-independent. It wasn't there before, right? It now lists HPV-independent cervical cancer as a subset. I'm not claiming this because of me, but I would like to think that some of the work that we did contributed in part to this new classification. The other thing I would like to mention is the work briefly that we've done on Ugandan cervical carcinomas. I put that in there only to say I've worked on Norwegian samples, I've worked on US samples, I've worked on Ugandan samples. It's time I worked on Nigerian samples. So I'm really hoping that we can form collaborations where we can do some similar things like this. I want to end with a paper we just published. This paper is on breast cancer. It was published literally less than a month ago, where we looked at, using the methods I've described, we looked at host and microbe RNA and tried to understand relationships between them and how they might be different between black and white women. So what we did basically was we got the RNA, we did the analysis I previously described, we identified the microbe in the tumors. And then we now said, let's do what is called hierarchical clustering. Just look at how related are these microbes to one another across the tumor. So what I'm showing there, think of it as a family tree, right? But what we see is very interesting. We got 2 major clusters of these tumors. So tumors went into, very interestingly, if you look at this column, oh, I guess you're not seeing it. So the column with pink and yellow, you would see that group 1 is mostly the yellow group. Group 2 is mostly the pink group. The yellow group are African Americans, pink group are European Americans. So we're seeing differences by race in the types of microbes that are present. We identify what the microbes are. This is just published. I'm happy to send the link to anyone for the sake of time. This, we saw microbes that were associated with African Americans and the ones that were associated with Caucasians. We now did something that I think was really, really cool. So I'm going to walk you through this. It may be complicated. So what we did was, we said, here are all the genes in these tumors. Here are all the microbes in these tumors. We expect that genes will be more related to one another. We expect that microbes will be more related to one another. But we took all of that information and put it in one single table. Okay? And then we now did Spearman correlation analysis to put them in 1,000 families of the combination of the microbes and the genes. Then, like I said, we expected that we'll see things that are similar to cluster together, and that's exactly what we saw. After the 1,823 were genes only. Makes sense. And 8 and 5 were bacteria only. But we saw about 172 that had at least one human and at least one bacterial piece of data. But because we thought, there might be spurious things in here. So we said, let's be very stringent. First of all, any data that was not complete and had less than 50%, we removed it. Then we used a correlation of 0.7. It was less than the correlation of 0.7, we also removed it. So we're down to 7 clusters, right? And then we looked at those clusters and we saw some interesting things. One of the things we saw is this example on your left. Just ignore the example on the right. Just look at the example on the left. So what you're looking at here is part of one of those 1,000 clusters. Remember, these clusters started out with some bacteria, some human genes. And we saw this interesting thing, where as you can see in green, Hafnia is a bacterium. PD2, the human gene. They were more closely related to one another than their fellow bacteria or their fellow genes. And we saw something that was also interesting. This gene and this bacterium had, when high levels, was associated with worse prognosis. So we're currently trying to understand the role of this gene and how it's related. We're also looking across the spectrum at other diseases and other cancers to see if we can find similar things. And so with that, I want to conclude by summarizing that we have discovered novel driver mutations in cervical cancer. We have established relationships between HPV integration and gene expression. I didn't talk much about some of the experiments, but we found some interesting data, and we did some drug analysis that suggests that you can use some drug combinations for the tumors that have MAPK1 mutations. And we have characterized HPV-independent cervical cancers, and we have identified microbes and hosts and their relationships in triple-negative breast cancer. And I would like to just acknowledge all the people who have contributed in some way. This is a long list over many years. And even in my lab, this is a long list, I would say, since 2014. So some people are no longer in the lab, but it's good to acknowledge the work that they've done. Then our funding sources, and with that, I'll be happy to questions. Thank you very much.
Wow. Wow. I just want us to give a thunderous applause to Dr. Akiemi Ojeshino, who has shown us today that our days in the basic medical sciences and biochemistry are not in vain. When we add them to our clinical sciences and public health, we produce fantastic results. A round of applause one more time. We may be seated. You may be seated, sir. So, Dr. Akiemi Ojeshino is glad to take our questions, but we will not be taking the questions here. We're going to plead that you will find him on LinkedIn. You will also find him with his email address that I hope he will graciously grant us today. And you will be able to directly ask him your questions, and he will be very glad to answer them. I want to gladly welcome the Deputy Vice Chancellor, Academics, Professor Juwon Arotiba. You're welcome, sir. Sir, you have a seat with us here, sir. You're welcome, sir, and we're glad that you made time out of this, your very tight schedule, to join us here today. We're also very grateful to all our teachers, our professors that are here, our former provost, Professor Akinka Omigwadu, all the professors, Professor Ogunbi, Professor Aimaku, all our HODs, Dr. Ayu, we cite you all. You're welcome, sirs and Maz. We also want to specially welcome, please do not forgive my bias, our retired professor of pediatric nephrology, who also made that time to join us today, Professor Adanze Asinobi. You're welcome, ma'am. We welcome all our brilliant, intelligent, first-class medical students. We're glad you're here. We welcome our dear nurses in making. We're glad to see you. Thank you for coming. Quickly, you will welcome with me. I am sure you now know that I was not just making any mouth or anything. You will welcome with me the Dean of the Faculty of Clinical Sciences, one major factor among many others that made this happen today, Professor Lawal, to make a presentation to our guests. Lecturer.
Deputy Vice Chancellor, Academic, the Provost of the College of Medicine, distinguished ladies and gentlemen. On behalf of this entire staff and students of the Faculty of Clinical Sciences of the College of Medicine, University of Ibadan, I present this to Dr. Akinyemi Ojeshino, Assistant Professor of Obstetrics and Gynecology at the Medical College of Wisconsin, Milwaukee, United States of America, as a mark of honor for delivering the special guest lecture of the Faculty of Clinical Sciences, the University of Ibadan. Today, the 3rd September 2025, this faculty recognizes your outstanding contribution to teaching, research, and especially an erudite delivery. Hearty congratulations and thank you so much. Hold on, we also want to give you this for you to remember us when you've done it, all right? Thank you so much. Accept it as a token from us.
Thank you very much, sir. When the guest lecturer was speaking, he did wonder if we had the press here. We have a very fantastic medical reporter who we're always glad to see, and we know that every information you've given here today is going to get to the right places. Please let's appreciate Mrs. Shade Ogutola from the Tribune. Right away, please welcome with me our faculty officer in the Faculty of Clinical Sciences, Mr. KK Adeogun, to give the vote of thanks.
Thank you, ma. Ní gbogbo ọ̀nà ẹ ṣé. Ní gbogbo ọ̀nà ẹ ṣé o bàbá. Deputy Vice Chancellor Academic, the Provost College of Medicine, Dean of the Faculty of Clinical Science, my Dean, my Secretary to the College, my own Oga, other deans present, former Provosts, medical students, nurses here present. We thank everybody for making this a successful outing. We pray the Almighty God to be with all of us now and forevermore.
Thank you, thank you, thank you. I didn't know our faculty officer could sing. Oh. All right. Will please take group photographs right away. And for the group photographs, we'll just like to invite all our teachers and professors present to join us right here. Yes, sir. Professor Omiwadu, Professor Gumbi, Professor Imaku. Please join us. Every oncologist present, actually. Every oncologist present. Please, we want to also appreciate all our students from physiotherapy who are here. Thank you for coming. Please. Okay. Okay. Dr. Ojeshino, sir, we will please plead that you just take a seat there so that our students can also take a picture. Please just stay seated first. May I request that students please come together, fill all the seats right from that second row. Please, the faculty present, please note that you are expected to have a tete-a-tete with the guest lecturer at the AOK Johnson boardroom immediately after this. The faculty present. So medical students, please move forward. Nursing students, everybody, you're here. So the rest of us can be on the aisles, the people at the back. We need you to fill the seats, please. Fill every seat in front of you. The people in pediatrics that I have taught, they know that. No empty seat in front of you. No empty seats in front of you. Physiotherapy students, come in. Nursing students, please come in. You are here and you should show that you are here. Please fill every seat in front of you, and then the rest at the back can stand. Move in, move in, move in. Yes. From that young man that is standing with the lady in blue and red, everybody should stand. Everybody behind them should stand. From that row, there are still seats in front, please. If you are not comfortable standing at the back, you can stand by the side. Thank you very much. Shall we rise for the anthem as we close today's meeting? Okay, shall we rise for the anthem?